For our first video of Section C, Interaction and Interdependence, we're going to look at enzymes and metabolism. Now, we've already discussed enzymes and proteins a little bit, and now we'll delve into a little bit deeper the characteristics that make up enzymes, some of their functions, and more specifically, things that can impact enzymes and their abilities to function. Enzymes function as a catalyst, and a catalyst is a substance that increases the rate of a chemical reaction, but it's not used up or changed or consumed during that process. And so it can be used over and over, and thereby only a small amount of these catalysts, these enzymes are needed. And enzymes are a biological catalyst, and so the substrates are altered by an enzyme to produce some sort of product. The substrate being what goes in, and the product then is what comes out from that reaction. These chemical reactions that enzymes help to facilitate, they can actually happen in life without an enzyme, but the enzyme helps to reduce the energy that's necessary, and so it would be extremely, extremely slow if the enzymes weren't functioning and weren't helping to carry out the reactions. Metabolism is a complex series of interdependent chemical reactions that occur in living organisms, and usually these are occurring inside of cells. Metabolic reactions form pathways in which the product of one molecule is changed into another via a number of different steps. Sometimes these are linear, sometimes they're cycles in nature, but it's the changing of one input into a different overall product. And metabolic reactions are catalyzed by enzymes. And so these enzymes can function as a series of steps that help to take an input and change it into a different output. And enzymes are what are doing the catalyzing or the changing of these specific reactions. And so there might be multiple or many different enzymes that are involved in this overall cycle, this sequence. Enzymes are specific. They are used in one specific reaction, and so they react with one input, one substrate, and that's specificity. We call it is often based off of the structure of the enzyme as well as the substrate, the input, in addition to the polarity of both the enzyme itself and the substrate. And this allows the control of how quickly the metabolic rate takes place or the rate of metabolism. It also allows for, as we'll see, for this metabolic pathway to be able to be stopped or paused temporarily, as we'll discuss a little bit later. And I like to think of metabolic pathways as kind of similar to a Rube Goldberg machine, if you've ever seen one of those, where maybe you drop a marble and it starts a reaction that hits something else and causes something else to happen. And so it's a series of multiple steps, and those steps in this case in metabolism are all carried out by different enzymes. There's two types of overall reactions that can take place. The first being anabolic reactions, and this is the buildup of smaller molecules into a larger molecule, so small to large. It does require energy. Production of macromolecules from monomers, usually using ATP, would be usually a solid example of what's taking place. More specific examples would be like in photosynthesis, the use of carbon dioxide and water, combining those with some sunlight to be able to produce sugar, the synthesis of DNA, taking small nucleotides and putting them together to make a DNA molecule, protein synthesis, making proteins, the synthesis of starch, cellulose, glycogen. All four of these, what they have in common is that small molecules, monomers, are taken and put together to be able to make a larger molecule. The second example is called a catabolic reaction, and this is essentially the opposite of anabolic, where it's the breakdown of larger molecules into smaller molecules, and this releases energy. Usually the reaction is coupled to the production of ATP. Some examples of this would include like the digestion of food, when we're eating food, the breakdown of those macromolecules into the individual monomers. A second example would be cell respiration and the use of glucose and oxygen to be able to produce ATP, as well as carbon dioxide and water. And lastly, decomposers breaking down dead organic matter, complex carbon compounds to release simple monomers as well. As we previously have established, enzymes are types of proteins, but enzymes specifically are globular proteins that have specific 3D structure and chemical properties, and both of these are really important for them to be able to function. The substrates, the input to be used with an enzyme, must be able to bind to a region on the enzyme called an active site. in which the shape and the chemical properties must match. Otherwise, they won't be able to bind with one another. While that substrate is bound, it's converted into the product that then is released, allowing the enzyme to catalyze the reaction again, so to be used again. Active sites themselves within the enzyme is where the location of where the substrate binds, and these vary in size, but usually it's only a few amino acids that must be attached to the active site in order to catalyze the reaction. Within the enzyme itself, the amino acids may not necessarily be physically next to one another, and they're brought together by the folding of the polypeptide within the protein to be able to catalyze that reaction within the enzyme. And then lastly, the alteration of the active sites can happen with changes in temperature or pH, and these can sometimes help, but oftentimes they have some pretty big negative impacts in allowing the reaction to be able to take place, and we'll look at these a little bit further on later in this video. The binding between the enzyme and the substrate is really pretty random. The substrate movement and the enzyme itself is random. Molecules are always moving. And so the binding between these two only occurs when they're close enough to one another. And when specifically the substrate is close enough to the enzyme's active site. And as we established, the chemical properties and the 3D properties, the shape, allow that specific substrate to attach to that specific enzyme. And so the substrate binds to the active site. It used to be thought of as a lock and a key, where the key would be the substrate binding or connecting to the enzyme. It doesn't exactly work like that, because the interactions between the two cause both the substrate and the active site to change slightly to one another, and it slightly alters the 3D shape, the structure of both the enzyme and the active site. And so a better explanation or phrase to be able to describe this is induced fit. When the substrate and the enzyme come together at the active site, it induces a fit between the two of them. Now, this doesn't mean that any substrate can bind with any enzyme. It's still dependent on those physical and chemical properties, but it just means that the two kind of mold to one another would be a good way to think about it. Sometimes some enzymes will have a second substrate, so after the first binds, the second would as well. And then changes to the substrate cause bonds to be broken and new bonds to form, and thereby converting the substrate into a new product. That product detaches from the enzyme, and the enzyme active site returns to its original state, and it's ready for the next substrate to be able to bind, thereby repeating the process. The binding of a substrate and an active site is a result of molecular motion, the movement of the substrate and the enzymes. Movement occurs in a fluid, the cytoplasm, the matrix, etc. And the molecules within this fluid are constantly changing directions and randomly moving in different directions. And so this results in random collisions. Those molecules are always moving around within that fluid, and they do so randomly. The rate of the collisions, when they actually connect, can change based off of increased amounts of substrates or enzymes, as well as temperature. Temperature can change the rate of how quickly molecules move. Typically, warmer temperatures result in faster movement. Regardless, the successful binding is dependent on the proximity, them being close to one another, the substrate to the active sites being close enough that they can actually bind. And so the movements and the collision that happens as a result is all random. There's variation within the movement and the motion of these different particles within the cytoplasm. The substrate and the enzyme are both in liquid, and so they can move freely and easily. If the substrate is large, it has less movement, it's limited, and so really the binding is dependent on the enzyme movement. A good example of this would be in DNA replication. The DNA is really large, so the enzymes are the ones that are moving around. Embedded or immobilized enzymes obviously cannot move around, and so the substrate is what's doing all of the moving. And then cytoplasm-based reactions are generally easier to take place because of the free movement of both particles compared to a large substrate or an immobilized enzyme. The cytoplasm reactions are easier to take place because of the free movement. Some examples of enzyme and substrate specificity, as we've talked about this process. A good example would be glucose binding to glucokinase. which is the enzyme that then is going to bind with glucose as that substrate. And so that's very specific. Glucose being a specific molecule binds with this specific enzyme. Hexokinase is a different enzyme, and while it's specific in binding with a hexose sugar, it's broad in the sense that it can bind with any hexose sugar, and so there would be different options of substrates that could bind with that, but it's not going to bind with other molecules. The structure of the enzyme is dependent on the amino acid interactions within the protein, both hydrophobic as well as hydrogen bonds, and the structure of the enzyme can be influenced by heat and acidity, can be easily changed, so small changes actually can have a big impact and make that enzyme ineffective as a catalyst, and changes that become too great or too big and cannot be reversed result in the denaturing of that enzyme, of that protein, essentially, which means that the enzyme breaks apart and it can't be used any longer. And so temperature can have a big impact on the enzyme itself. Heated liquid that the enzyme would be in, as well as the substrate, when it's heated, the particles gain kinetic energy, and that then results in the enzyme and the substrates moving around faster, as we previously mentioned, and it increases the chances of collision, which thereby increases the enzyme activity. This increased temperature causes more and more vibration within the bonds of the enzyme, and so if it increases, the chance of them breaking also increases, and this can then lead to the protein, the enzyme, being denatured and broken apart and not being able to be used again. The different types of enzymes have different points of being denatured, so different enzymes have different temperature ranges that they're stable or they can tolerate. But temperature can have a big impact on changing the rate of enzyme activity up until the point of it becoming denatured. pH is a second example that can influence enzymes. Acidity is affected by the presence of hydrogen ions, so an acid has more ions, makes it more acidic, and pH is measured on a scale of 1 to 14, and it's a logarithmic scale, meaning that if you're increasing or decreasing by 1 unit, such as from 7 to 8, that's not increasing it by 1, it is actually, the solution is 10 times more acidic or basic. And within that scale, 7 is neutral, numbers lower than 7 are acidic, and higher than 7 are basic. And enzymes have an optimum pH value, and if it changes, if the pH level changes above or below that, it can result in the change to the structure of the enzyme, making it less and less effective until it becomes denatured as well. So similar to temperature, changes in pH can also result in the denaturing of that enzyme. Not all enzymes have the same optimum pH, just like temperature, and those pH tolerance levels vary due to varied environments depending on the specific organism. Substrate concentration can also influence the rate of enzyme activity, but in this case we see it not so much be a negative as the collision between enzyme and substrate is random in liquids. More substrate is going to increase the chances of collision and thus increase the reaction rate. But this is not an indefinite exponential increase. As more and more substrate is added, assuming that the amount of enzymes stays the same, eventually all of those enzymes will be occupied. And so at that point, then it becomes saturated and the reaction rate eventually plateaus. It can't go any faster because there's just not enough enzymes to be able to process all of the substrates. So it increases and then level off after it becomes saturated. The reaction rate of an enzyme reaction is the speed at which the substances are converted into products. A couple of different ways that this can be measured. You can allow the reaction to happen for a fixed period of time and then measure the amount of substrates used or produced. You could also start with a known amount of substrate and continue the reaction until all of it is converted to products, and then measure the time period. One thing that I also like to use with enzyme reactions within the classroom is to use veneer probes. They make a number of different scientific probes that can be used, gas pressure, carbon dioxide, oxygen, and then those gases can be measured and tracked over a time period to assess the rate of enzyme activity. Highly recommend using those if you're trying to do this for an IA or for an investigation.